Wow. It's a great privilege and pleasure for me to introduce Dr. James Watson to you this afternoon. As Dan said, he's a guest of the Cancer Center, and I think we're all very excited, obviously, that he's here to share his ideas with us. Let me start with what you all know about Jim. You know that it was his 1953 paper with Francis Crick that unleashed the revolution in biology of the 20th century, with all its ramifications for biomedicine, for agriculture, for biology, for forensics, et cetera, et cetera, et cetera. You probably also know that Jim is a writer of important textbooks, communicating what's important to younger people. And he has also written a number of historical views of the scientific process, some of which some of you may have read. You may also know that when Jim took over the Cold Spring Harbor Laboratory on Long Island in 1968, it was a sort of sleepy little lab, and he transformed it into one of the world's leading centers for biological research. And you know that the lab has turned out to be a mecca, both for scientific meetings and for courses. And you know that it's also contributed as a sort of world-class research institution to research related to cancer and more recently related to neurobiology. As Dan mentioned, I first knew Jim well as a graduate student in his lab in the mid-1960s. So Jim was professor of biology at Harvard from 1956 to 1976. Even though he had taken over the Cold Spring Harbor lab in 1968, he did both of them for a while. And what I want to do is relate to you a couple of anecdotes from that time. And I want to start with this slide. So during the time that Jim had a research group at Harvard, every summer, is there a pointer? Ooh, I didn't ask about that. I guess there isn't a pointer. We would gather together and assemble on one of the rhinoceros statues in front of the biolabs at Harvard and get a group picture taken. So you see second from the left in the front row is Jim. The next person is Wally Gilbert, who by that time was sort of co-sharing running the lab with Jim. Two people over sitting in the chair is Mario Capecchi, a Nobel Prize winner, now professor at Utah. And if you go up into the second row, the second person from the right is Peter Moore, a longtime professor of chemistry here at Yale. During the summers, the number of the Watson group always sort of blossomed a little bit, increased a little bit, because Jim invariably got a lot of undergraduate science students to come into the lab. These were mostly from Radcliffe. And he assigned these people, you see them in the top row there, to work as technicians for the advanced graduate students and postdocs in the lab. And it was Jim's fervent conviction that if you had somebody to whom you had to assign work, you would probably get more work done yourself. And that was why he did it. Let me tell you, he'll need that, yeah. Let me tell you just two other stories that I recall from when I was in Jim's lab. One was about a lab outing to the White Mountains. So we had persuaded Jim that he should come on a hiking trip up to the Presidential Range in the White Mountains. And we arrived in the late morning, having driven up from Boston at the trailhead. And we started up the trail. And Jim started out very, very slowly and deliberately, one step at a time up this trail. And those of us who were the graduate students, 20-somethings, very eager, very avid, dashed on ahead, and then, of course, had to stop and take a breath and dashed on ahead again. By the time we finally arrived at the Lake of the Clouds hut, some 3 or 4 hours later, there was Jim lying in front of the hut in the sunshine where he had been for about an hour, just proving that Jim always knows where he's going and he knows how to get there. The last story I want to relate to you is the one about the lady in the box. 1966 was Francis Crick's 50th birthday. And Jim decided, and there was to be a celebration at a Cold Spring Harbor meeting. And Jim decided that what Francis needed for his birthday was a very large box out of which would pop at the appropriate moment a lady, scantily clad, of course. Now, Jim had to find somebody who was going to provide this box, but New York City provides everything. So he was able to find a purveyor of such boxes and went down to meet with the people in charge in Manhattan. And he was apparently confronted with a very large sort of scrapbook, sort of a rogues gallery of pictures of all the possible ladies that might pop out of the box. And apparently this went on for quite some time, and finally the agency manager says to Jim, "Look, doctor, you know your business, and I know my business, and you want this one." And so the lady that popped out of the box was in fact that one, and the whole thing was a very, very great success. So since I know that you've come to hear Jim, not me, I'd like to turn over the podium to Jim, and I have promised to put his first slide on. Whoops, should be maybe the next one. There we go.
Yeah. So the title of my talk was called Curing Incurable Cancer. And I've given the same talk about, 5 times, so I can't, I don't want to give it again. And so I've also given a couple times a talk called Driven by Ideas, which I gave in front of the geneticists in Seattle, and I didn't talk about cancer. So, but this is a combination of the two. Of course, I've been driven by curiosity, but I think as I got older, I've been more driven by, through reading or something, thinking I understand something, and then I want to know whether, which is sort of an idea. You explain something, and so at times I got very excited by an idea. So I can't even sleep, and now I have a few ideas about cancer, which actually have, on some nights, made me hard to sleep. And I should say my interest in cancer goes back a long time. I first heard of cancer in a sort of scientific way when I took Salvador Luria's course on viruses at Indiana University in the fall of 1947. And it was a semester, and toward the end of it, he talked about tumor viruses, which were very mysterious. But, tobacco mosaic virus, they could even be crystallized, and in just that time, my father's younger brother had been diagnosed with malignant melanoma and died soon after the course finished. So I've always had a desire, and to sometimes first understand cancer and then cure it. Malignant melanoma is awful. And so my sort of intense interest in cancer is, I gave a course on cancer at Harvard before I met Joan. in 1959. And so, you know, I had 6 students, and there wasn't much to say. And there was really only one, there were a couple of things, but the chief was Warburg, and the cancer cells secreted lots of lactic acid and had high glycolysis. And that was the sort of dominated cancer research. And then genes have taken over to the point of where most people doing cancer research couldn't pass the biochemistry exam. That is, they've been so dominated by, you know, genes are so many of them, and the whole thing is that, you know, molecular biologists don't know biochemistry. I mean, that's, or most, I mean, in Yale, you know, you got more balance, but if you really look at it, the ignorance of biochemistry among most of the people, leaders of American cancer research, is abysmal. Okay. Yeah. And so what is needed now is, you know, more biochemistry, and I think you'll see that by the end of my talk. Okay, now if I, let's see. Yeah, I think this is, yeah, this is the one. Okay, all right. So the first idea, I just want to, I'll go back to the DNA, is these were the A form I saw in Naples in May of 1951. Wilkins had just taken it, and I got enormously excited, and you know, once I saw that photograph, I'd heard of X-ray photographs for DNA at Indiana University. Felix Horowitz taught a good biochemistry course, and he talked about, you know, Perutz and Burnell and Asbury, had taken X-ray photographs for DNA in the late 30s. But they were fuzzy, and this picture was really very sharp, unlike Asbury's pictures, and Wilkins said it was crystalline, and that meant DNA had to have a structure. The second form was the B form, which I only saw a year and a half afterwards, and that the cross said it was the helix. But the second idea was, and the idea here really wasn't mine, was a paper by Linus Pauling and Max Delbruck written in 1940, which said that the way genes would be copied would probably involve complementarity, that is, positive attracts negative, essentially. So you would copy a biological molecule by making its complement. And there had a paper appeared before by a very good physicist named Pascal Jordan, who thought he had a scheme for like-by-like copying, that quantum theory would allow you to copy something identical. But Pauling thought that was wrong and told Delbruck to put his name on the paper, and so this paper appeared. To my great, and then Pauling gave a talk in Manchester, probably in 47, in which he repeated it. Then luckily he forgot his own idea. And you know, what good luck. But the idea was that, and that was my idea, that the pairing between impurine and pyrimidine, and that came the morning of February 28, 1953. And I wasn't trying to do complementary at all. I knew it, but I just wanted to get the right thing for the structure. And it turned out it was complementary. And that was too good to be true. And there's a picture of Shargaf who had the data, and I didn't, the A and T was pretty good, but the G and C wasn't really equal, really because the analytical methods were not good. And then I met Shargaf, and I formed an intense dislike of him and decided I didn't want to use his data. Yeah, so personalities do enter science. And Francis Crick, actually, we met Shargaf, and Francis took the idea seriously and knew a young chemist, John Griffith, who thought that A and T would attract each other not by hydrogen bonds, but by sort of quantum forces, so they'd be on top of each other. And Francis tried to do experiments of changing the spectral changes by mixing A and T together in G and C. Didn't get anything, forgot about it. And then I only got them, and I won't tell the story, but it's in the double helix. When I was told that, guanine and cytosine, guanine and thymine had enol structures instead of keto, and the base pairs just fell out of that at all. It was not really thinking. There was and there's the picture of Francis and I. Interestingly, it happened because someone tried to sell a story to Time magazine that they should report the double helix, and they sent someone over, Barrington Brown, who took the photograph, and he was paid about $2 for the photograph. And then it was never used or seen by anyone until I wrote the book, The Double Helix, and included it. Then he had a good business of selling them. He was quite a clever person. He had, as an assistant helping him, Princess Margaret's future husband, the photographer and designer. Anyways, he was a good guy. Now, this is an idea that was sort of obvious, that when we started looking at phages and realizing, there was a life cycle, that the timing as to when the molecules appeared was dictated by their order along the DNA molecule, and that basically transcription went along at about 60 nucleotides per second, roughly that. And so if you were transcribing a molecule, you could say, well, that molecule won't be ready for 5 minutes or something like that. And so the whole cycle was explained by that. And that didn't make me, I didn't get really excited until a couple years later, Dick Burgess discovered the sigma transcription factor, and I thought that you could time more complicated things by, you would make a transcription factor that would take a certain number of minutes to make the first one, and then that would start the second synthesis, and so you could, say, time a bacterial life cycle quite easily just by the transcription of times of adding together several genes, and you would end up with the right thing. So I published these, in the Molecular Virology, the gene. If I had an idea, and that meant you never had, it was a wonderful way you could say the idea, and there were no referees. Yeah. Now often, you have people won't publish you because they say your idea isn't proven. Whereas I think, when you teach, something is 90% right, just treat it as 100%. And then, then in your next course you learn the exceptions. But in the initial course, you get it as simple as you can, and then, with time. So I like this. and then thought of this scheme by which sequential synthesis could do it, okay? And then in the fourth edition, this was in the second. In the fourth, there's this one. Splicing had been discovered, and people had thought, what are introns for? And introns are there to make possible long life. Okay. So if you have something that you want to separate by an hour, just make a long RNA molecule. Okay. So this is this edition is, I think, probably the best edition after the first, really because I managed to get Nancy Hopkins, Jeff Roberts, Joan Stites, and Alan Reiner to do it. So it was half a Yale book. So it was a very good one. But so I had this idea, but then was it right? And so Mike Levine took over and wrote the next two editions and talked about the chief problem of, in development is pattern formation. And they left out any mention of timing. They took out this paragraph. So Mike was just in Cold Spring Harbor, and I said, what about timing? And he said, you're right, that if you take really short-lived organisms, I mean, eukaryotic, okay, which have splicing, the introns are very small, and then if you take a related but one of the longest banded, the introns are longer. So I think no one has actually, experimentally taken an intron, changed its length, and shown, mistiming. And there's lots of different genes that you could use for it. But I think it explains why they exist. And, bacteria don't need introns, so they don't have them. Simple. Anyways, that's the thought, and it's been totally ignored by the whole biological world. No one's asked how you time things. And Seymour Benzer, they came up with the clock genes, but they don't provide a metronome. Okay. So I think that's actually a real good idea. Now here's one which has got me into trouble, but it's a really good idea. Okay, so as you can read here, I went out to a meeting in Los Angeles to be on gene therapy, and so they gave me a driver. It turned out to take me to UCLA, and I asked what he was doing, and he said he was trying to chemically modify melanophore-stimulating hormone to give it a longer half-life, so you could inject it and get tan without going into the sun. So it was an anti-melanoma device, okay? And he thought of it, a good market in Australia. Okay, so they synthesized one with a couple of unusual amino acids, so it wasn't clipped down and had longer life. And he, in the right experiment, injected it himself. And he told me he got an 8.5-hour erection. Okay. So that really, this was just before Viagra had appeared, okay? So he was having hopes of, becoming rich, but it has to be taken by injection, so it's, okay. And this was, just the chemistry of the sort of thing. And, but this is, if you think about it, there was what men call gallantry and God's adultery is much more common when the climate's sultry. So that there's a connection between sex and sun, okay? And you could say, so what? But then, if you looked at the gene, it was extremely interesting. I would have never cared what was the structure of pro-opiomelanocortin. And so through gene sequencing, you found that MSH was made as part of a longer protein, in which the MSH, the amino end of the carboxy end, is endorphins. So when you go in the sun, you feel both sexy and happy. Okay? And that's good, because you know, going in the sun, you need to do it to make vitamin D, and you're rewarded. So you've got to ask, you know, there are no Bible classes for animals, so how do you learn how to behave? And so you get a reward for good behavior, and it's all right. It's pretty straightforward. But the interesting thing is this molecule, there are other receptors. You know, one turns on melanin synthesis. The other is this molecule is turned up by leptins. So leptin come, and they're made in fat cells. And so when you eat, you're happy. Okay? So there's a reward for happy, and it comes in two phases. So, but the consequence of it, you know, leptin, but people who are very thin are very unhappy. And so supermodels essentially have been associated with drug use, yes. They're too thin to be happy. Okay. And so here's one, you know. Let me have men about me that are fat, sleek-headed men, and such as sleep o' nights. Young Cassius has a young and hungry look. He thinks too much, such men are dangerous. Okay, so this has sort of been known in a long time that, you know, heavy people are more content than thin people. So naturally, you know, when you seek out graduate students, you want thin ones. Yeah. Yeah, you don't want people too content. Okay. And there is a sort of life cycle. You think if people start out thin, they work hard, and then in middle age, they become heavy. Yeah. So, you know, and essentially the function of old people is just to give advice to young people who do all the work, you know, on their pensions and so on. Okay. And this is a joke, you know, no woman can be too rich or too thin. You know, sounds like, you know, a description of the Duchess of Windsor. But when I gave this talk at Berkeley, some people were very upset. And so you can follow, of course, that, you know, bald men are more attractive to women because they get more sun on their head, okay? So that got me into a lot of trouble because too many of the people who run science at Berkeley are too thin for their own good. Now, just to go back, that and this idea is someone over in the Harvard Medical School has discovered, you know, endorphins in sun, but he's never really written it up, and I'm finally going to write it up. But interesting, the opioid receptors are concentrated in an organ called the habenula, which fires when you're frustrated. And so they're normally, the endorphins go basically turn up dopamine synthesis and serotonin. Yeah. So endorphins are extremely important. And so they should be rewarded as, you know, you do the right thing for your body by avoiding being thin. Yeah, okay. So this is one of my better ideas. It's never been, I had it 10 years ago, and this newspaper article put me off writing it up, and I'm going to write it up because it explains something. It explains a lot. And Okay, then here's my first idea about cancer. I was teaching this course. No, I was teaching the virus course. It was before I gave the cancer course. And I was going to have to talk about tumor viruses. I suddenly, I had learned about 2 months before that the bacterial viruses, T24, actually code for an enzyme which makes hydroxymethylcytosine. I'd always thought of viruses as the proteins were just not enzymatic, but sort of structural. They're just a container to carry the virus from one cell to another, and never had thought of viruses as coding for enzymes to modify the metabolism of, or to in some way make the cell replicate the virus. So this got me extremely excited. I thought they had to carry a gene. It turned out to be right, and was probably, so I put it in the molecular biology of the gene, and it formed the basis of the first chapter. And these were, if you looked at transformed cells, they contained a protein that wasn't present in normal, and that had to be, I thought it had to be coded for by the virus. So the idea, it led me first to go to Mass General for the summer and work with papillomaviruses, but there was, not a good biological system. It was the development of the polyomavirus system, and then SV40 being discovered, and yeah, it wasn't really proven, we didn't find for these viruses, it wasn't a simple oncogene. The T antigen bound a tumor suppressor. These were concepts we didn't have, but the, T antigen binds tumor suppressor, so tumor suppressor, the cancer takes off. And Ed Harlow had done that in, it was 89. So the idea wasn't, it was from 59 to 89. But it drove me, drove me to, because, I had the advantage, if you have an idea, then you are more motivated than people who are just doing it because they want to study cancer but don't have any idea. So the idea really keeps you up. Okay, now this is the first idea I'll say here, which I really only had thought this way about 3 months ago. And the problem which I had been sort of posing was, why does chemotherapy preferentially kill cancer cells? And it turns out that cancer, and I'll give some evidence for this, it's much easier to kill a cancer cell than a normal cell. And so this is always described, but I never read anyone say, well, why? Well, I think it is, in a very simple way, that the essence of cancer is the genes turned on that promote growth and cell proliferation. And so they're always on. Whereas a cell will normally go on and off for growing, cancer cells just like to grow too much. And so, if you add, inhibitors of ubiquitin system, or if you stop autophagy, the cancer cell preferentially dies. So the cancer cell is just making too much. The metabolism is going too fast, and particularly the glycolytic metabolism. That is, growing cells have to glycolyze to make the amino acids, the sugars, etc. You can't, a cell has to go through, when I say glycolysis, also, it's glucose and glutamine, so the two of them. So, all right. So this was, if I sort of wrote this sort of thing, it was rejected by an awful editor. And, I think that the trouble is that people, they don't want to publish anything wrong, and I don't think it matters if the idea is wrong, if it motivates you to do experiments, and it just might be right. But so everything I put in my textbooks, these sort of concept types, I thought they're for the most part right. I think there was only one out of, probably, several hundred that turned out to be wrong. But, there were always, 95% exceptions, but just have the big one, okay? So just, and what it is don't think of the cancer cell as Superman, but as a sick man, okay? The cell itself is sick. So it's easy to knock off if you can do it the right way. You just shouldn't do it the wrong way, okay? And we're, for the most part, doing it the wrong way. Okay, so this is essentially same thing. So the cancer cells are sort of constitutively on for glycolysis, and if you really, you can sort of, it leads to preference in two ways. The sudden disruption of ATP, so that goes down to zero, and you can do it by 3-bromopyruvate, preferentially kills cancer cells. And then metformin-activated AMP kinase preferentially kills the most glycolytic cells. Okay. And here's the 3-bromopyruvate. It's never been used on humans. It's been used on rats by only one person, and it basically cured rats with very big liver cancers. They all disappeared. They injected the material right into the tumor. Okay. So the tumors couldn't take having a sudden depletion of ATP. Okay. It's also a pretty toxic molecule, and so it's never even been in phase 1 human tests. Then there's metformin. And metformin is the most commonly prescribed drug in the world. It's taken by people with type 2 diabetes. And about 5 years ago, it was published, and here was the paper from Dundee, that there's a lower cancer incidence in people with who have diabetes. Now, there are two possible explanations. One is that if you take that, the metformin will lower insulin levels, and insulin actually is a driver of many cancers if you take it when it binds the receptor. And so all you're lowering, you're stopping the cancer by lowering insulin. The other is that the metformin works by directly killing the cancer cell. It could work in both ways. Okay? And you can kill cells preferentially by metformin outside of animals, so insulin isn't involved. Okay. So the insulin may explain much of the result, but there's also a separate one. Okay. And metformin is supposed to essentially, metabolic stress is when you run out of energy, or you run out of things you need to grow. And when you do that, the cell's response is just to stop going into cell division. You don't want to make any, if you want to essentially conserve everything, so you shut it down. And Craig Thompson, and I'll show the data in a moment, most interestingly said that metformin is most effective against cancer cells when they're p53 minus. So that's very important, because generally, if the general thought is if you lost p53, your cancer is harder to control because the cancer is worse. But what I think it's doing is the cancer is becoming even sicker because it's running glycolysis at an even faster rate. And then if you do something to disrupt the energy supply, the cancer cell just dies. Kevin Struhl has published some works on yeast transcription, and a very good scientist, has published on metformin, which he found in just, as a screen, he screened all existing drugs, see those which would stop transformation, and metformin popped up at the top. And has published quite good data that metformin preferentially kills mesenchymal stem cells. That is, these are the worst of the cancer cells. The ones that exist with the tumor, and for the most part. And there I just raised that does exercise also slow down the rate of cancer, which there's pretty good evidence for metastatic prostate cancer just by lowering your ATP level. So when I try to play tennis about 3 times a week, and so I go through, I play against a pro who runs me, and at times I just can't run anymore. I just give up. And then I think my ATP level is really down, and it comes back up. So I think, extreme exercise really is subjecting you to a lot of metabolic stress. Yeah. So you probably shouldn't eat too much before your tennis match. You just want to get that stress. Anyways, I'm now taking, that's sort of the summary. So I think, as a fact, the good fact, metformin does stop a lot of cancer. And it essentially stops glycolysis. Okay. And okay. And so ever since I read this fact, which is trying to be confirmed with some tests, I take metformin every morning. Okay. And I play that, and this is out of a Craig Thompson thing, and it's very key that if you activate AMP kinase, then you turn on P53. If you can't, you no 50, and P53, basically stops glycolysis. So I should add, I guess, whether, from the viewpoint of a good biochemist, the mechanism of action of metformin is still a little messy. And whether it really acts through the AMP kinase, we don't know, but the good fact is that metformin is a good anti-cancer agent. Okay, I think that's, so I'm not worried about that fact being overturned. And this just shows you the real difference between P53 minus and normal. Metformin really goes at the on P53. I think that's a very important fact. But I've never read any paper or anyone getting excited by it except me. But then I worry, the whole field of cancer research never gets excited. It just accumulates more data. Yeah, because it should spend more time thinking and less time doing experiments. Okay, all right. And I think the trouble is there's so many facts. And I can sort of understand the trap we're in, but I think you should realize it. And, okay, so we'll end with this sort of, incurable late-stage cancers. And what are, effectively, the first is that the more aggressive cancers really are stem cell-like, and that's not at all surprising because stem cells are, cells which can divide. Okay, so so they're effectively, when I say dedifferentiated, they're not epithelial. That is, cancers largely start out as epithelial cells, carcinomas, but with time, they're more mesenchymal. The other is that, the Warburg, the highly glycolytic, they have a high self-digestion, which I think is a response. They're more vulnerable because everything is just set for too much synthesis. They tend to be resistant to targeted therapies as well as, we call them incurable because we can't do anything, so we don't have anything which will work. Okay? And they're hard to get to move into apoptosis. Now, item number 7 is one which has largely been ignored by everyone. And it is that late-stage cancer secretes large amounts of interleukin-6. This was the paper that they rejected me. I wanted to point attention to it. Okay? The observation has been known for breast cancer since 1990, so it's an old one. And so basically, cytokines, if you look at them, they often function just to turn on cell division, of the immune cells. So there's cell division promoting thing, and interleukin-6 turns on STAT3, and STAT3 turns on MYC. And so in lung cancer, you can essentially make them resistant to the targeted therapy by just adding interleukin-6. Okay. And I just saw an abstract from Cold Spring Harbor on breast cancer and HER2 breast cancer, which is pretty mesenchymal. When you look at it, large amounts of interleukin-6. And when they add antibody against interleukin-6, they slow down the growth of the cell. So a key component when you sort of go toward this late stage is you just have a cascade of cytokines. And there's lots of literature which just, pointing at, starting with N-kappa B, which, it's very complicated. But the moral is that if you want to reduce your chance of getting cancer, just take an anti-inflammatory. And certainly the aspirin is a very good way of lowering cancer incidence. I bleed a lot with aspirin, so I take ibuprofen. Okay. So that's, and I take as much as I can without bleeding. So, the aches and pains you get from being old, you can stop that through it. And, then I think I'm getting the added benefit of, Okay, and then this last thing, I think very important, well, this is sort of, sort of known if you look at it, that you bind to a IL-6 receptor, and then it goes through STAT3. STAT3 also turns off apoptosis. So people are trying drugs against STAT3. It's an obvious one to try against. Now, I think, And then here's an idea, much of the resistance to anti-cancer drugs that lead to incurable cancer reflects epigenetic changes that produce new drivers. So your old therapies don't work because you've got new drivers. Okay. So you won't find the epigenetic thing is not revealed by DNA sequencing. Okay. So DNA sequencing will, tell you the changes in the DNA, but they won't tell you the changes in chromatin that result from epigenetic things. Certainly in Bob Weinberg has written, very persuasively about EMTs, but I think, at least among some people, it's a sort of, they think the matter is not decided. But I think, now, a case where this may be true is Josie Schlesinger's wonderful anti-BRAF drug. You get resistance. And if you remove the drug, and Yossi may tell me I'm wrong, but Milan Herman told me that when you culture the resistant cells, they return to being normal, at least in his cases. So I think it's an epigenetic change. There's, you remove the selection, and they revert back to drug sensitive. But when you add the drug, they will go to the form that resist it. So we have to solve the resistant thing, the epigenetic one. Okay. That's sort of. Oh, this is, I got to it. And that was the main thing I want to say. So in the manuscript I had, which was rejected, I sent this to a friend, and he crossed out everything I wrote about cytokines. He said it wasn't proven. Okay. Now, he was one of my own students. But, I think he just risen in life and, grants and all that he stopped reading. So I think a good life, if you really, would be 1/3 experiments, 1/3 reading, and 1/3 exercise. But, if you don't read, you're really in problems, I think. And the literature is really large. Yeah. This is this result of Kevin's rule. The IL-6 really pushes you into being a good active stem cell. And this is just typical, this regulation of the cytokines. Okay, now this is a semi-big idea, which is that anti-cancer drugs targeted against cell proliferation, that is, say, MYC, will lead to less toxic side effects than anti-cancer drugs targeted against growth-promoting signals. So if you have a drug against PI3 kinase, it's going to have inherently lots of side effects, even though you're going through, specific ones. Because they function, your nerve cells can't function without constantly making new axons and dendrites. So growth is absolutely essential. So it's not surprising that these things make you feel punk. So don't try and stop growth. That's that would be my message. Go for cell proliferation. Now, this is what I'm saying is sort of obvious, and you could say, well, why haven't people thought this way? And I think the reason we just haven't had any drugs that specifically work against, that stop MYC or stop the cell cycle. But, So I'll just conclude with work done at Cold Spring Harbor by Jonathan Zuber and Chris Badish, who were trying to cure a form of leukemia which was essentially chemo-resistant. The drugs don't work against it. No drug works against it at all. And Cold Spring Harbor is one of the centers for RNAi development, and the idea was to use RNAi to knock out the genes to find out which are essential for the cancer. And Chris's big advance came when he decided that he would only knock out those concerned with epigenetics, which are 1% of the total genes, so you would only have to have 1,000 probes instead of 100,000 probes if you knocked out all the genes. So it's biased. It's only discover epigenetic chromatin modifications when they are the vulnerable point in the cell. So, and this, you've seen this slide here, or at least some of you, so I won't dwell on it, but it was the sort of work done at the level of, one person, not a big team. Okay. And then they discovered a protein which I had never heard of before, a protein involved in gene activation of bromodomain protein. Now, thanks to the Human Genome Project, we know there are 43 different bromodomain proteins. So the functioning we don't know. The lucky thing in this case, BRD4, is that it had been implicated in a rare form of cancer called midline carcinoma in a chromosomal translocation. And it produced a virulent childhood cancer which kills you in a couple of months, and that's it. So a drug had been made against BRD4, and when tested, and that is, that's sort of. And this was the paper by Jay Bradler working at the Dana-Farber Cancer Center in Boston. So he had a drug against it. And when, unlike other forms of treatment, this actually stopped the cancer. It didn't stop, it's not the optimum, they're developing a more advanced version of it. And it hasn't gone into humans, but the interesting thing is it didn't seem, at least, to be affecting normal hematopoiesis. So it may have very few side effects. I'll be very nervous until it gets into the first people, which hopefully will be in 6 months or so. But it sort of puts MYC at the center of it, and very good experiments were done by Gerard Evans in San Francisco, where he knocked out all MYC by a dominant negative plasmid, so no MYC, and 6 types of xenograft models against cancer. It worked on all of them, but when you knock out all of MYC, the stem cells stop dividing, and the skin gets thin. And so in his experiments, he would have the drug for 3 weeks, then 3 weeks without, back on. And in about 4 cycles, he seemed to have cured each one of these. If you'd say why, it's just under this sort of shock that the cancer cells are more vulnerable than the normal ones. You have to keep coming back to that you're going to be aided by this fact. What isn't known, and I think the big question is how many different ways you can turn on MYC. So will resistance develop to the drug because here's a selection of a new way of turning MYC on? So we're really just at the start, I think, of trying to understand chromatin. I've had a sort of hate relation to chromatin, you know, modifying histones, because I didn't know where it was going. But now, since it seems to be the heart of cancer, there's nothing but we can do we have to actually learn it. You know, and it's simple. Bromodomains turn off genes, bromodomains turn on genes, and methyl groups turn them off. That's simplified. But, so cancer, I mean, at least a lot of it will, I think our next couple of years will depend on understanding really chromosome remodeling finally much better. Okay, my last idea is not is sort of half-baked, but the fact that you can sort of lower the incidence of, you know, cancer prevention without any side effects. That is, metformin or, you know, vitamin D is claimed to, whether that's a good one, but I take vitamin D every day. There's some evidence. And that Judith Folkman's molecules, endostatin and Novak's thrombospondin, if you knock them out in mouse models, they get cancer faster. So they've actually been preventing cancer without any side effects. So it seems to me we just might be lucky enough that we will, you know, move to an era where much of cancer can be controlled the way we control bacterial infections, taking a medicine and without terrible side effects, and that there'll be no more bone marrow replacements. So we'll need, you know, less medicine and less doctors and less smaller hospitals. But that's an ideal view of the world. At least I think we have a chance to stop a lot of cancer by just trying to repeat what has been done. That is, instead of trying to inhibit the drivers, to just directly determine by RNAi the Achilles heels of different cancer cells. So find the Achilles heel and do it in an unbiased manner. Sort of outthinking the cancer cell will be very hard. So just, you know, don't, stop thinking and just do the RNA. That's sort of my thought. But you see, everyone has their own driver, and they just hope a drug against the driver will do it. So, but it won't be done if it's done at the level of, I mean, it won't be done fast enough unless we have semi-industrialized labs. So you could say, well, you know, form new biotech companies. Or the other is, why not do it at Yale? You know, which would mean, you know, a slightly different process, slightly bigger labs, short time. But, you know, if you say there's going to be a war in cancer, why not act like a war is on? Which means, you know, probably true bosses. You know, just, you know, just take the space and get it done. And, but it probably, you know, whether it'll work will certainly not cost $1 billion. I mean, the sums of money are pretty small. the Cancer Genome Atlas is costing us, I think, $100 million a year or something like that. And we should certainly be trying. That's to find new drivers, and I'm afraid we know most of the drivers already. But we don't know the Achilles heel. All right, I will stop at saying I'm having a lot of fun trying to solve cancer through thinking as opposed to experimenting. And but I don't think the cancer is too important not to be handled intelligently. Thank you.